Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today I'm going to quickly go over vitamins and then finish off with a day in the life of food. So vitamins are also absorbed in the small intestine. They can be categorized as either fat-soluble or water-soluble. Because there are only 4 fat-soluble vitamins, vitamins A, D, E, and K, these can be easily memorized. All other vitamins, B complex and C, are water-soluble. Fat-soluble vitamins dissolve directly into chylomicrons to enter the body. Failure to digest and absorb fat properly, which can be due to pathologies in the liver, gallbladders, pancreas, or small intestine, may lead to deficiencies of fat-soluble vitamins. The water-soluble vitamins are absorbed along with water, amino acids, and carbohydrates across the endothelial cells of the small intestine, passing directly into the plasma. And so let's talk about the function of each vitamin. So vitamin A, which is also known as retinol, is a visual pigment that changes conformation in response to light. And I talked more about this in the phototransduction cascade videos I posted earlier. Vitamin D stimulates calcium absorption from the gut, and it helps control calcium deposition in bones. Vitamin E prevents oxidation of unsaturated fats, and vitamin K is necessary for formation of blood coagulation factors. Moving on to water-soluble molecules. Vitamin B1, or thiamine, is needed for enzymatic decarboxylations. Vitamin B2, or riboflavin, is made into FAD, which is an electron transporter. Vitamin B3, or niacin, is made into NAD+, which is also an electron transporter. Vitamin B6, or cobalamin, is a coenzyme that is involved in the reduction of nucleotides to deoxynucleotides. Vitamin C, or ascorbic acid, is necessary for collagen formation, and deficiency of this results in scurvy. Then we have biotin, which is a prosthetic group essential for transport of carbon dioxide groups. And finally, we have folate, which is an enzyme cofactor used in the transport of methylene groups, synthesis of purines and thiamine, and required for normal fetal nervous system development. And so now let's talk about a day in the life of food. So imagine you purchase a sourdough baguette and tear off a piece. Chewing increases the bread surface area, allowing it to soak up more saliva. Now ptyalin, or salivary amylase, hydrolyzes starch into fragments, while the tongue and cheeks form a bolus. As you swallow, the upper esophageal sphincter relaxes, peristalsis carries the bolus to the stomach, the lower esophageal sphincter relaxes to let it pass. In the stomach, strong acid destroys most of the microorganisms which are present in the bread, while further hydrolyzing some polysaccharides. The stomach thoroughly churns the bread, forming acidic chyme, which is gradually released into the duodenum. In the duodenum, pancreatic amylase chops the polysaccharides into disaccharides. These diffuse into the intestinal brush border. Here, the disaccharides are hydrolyzed to monosaccharides. Digestion is complete. Up to this point, none of the sugar composing the bread has entered your body. It remains on the luminal side of the GI epithelial tight junctions. Since they are bulky and hydrophilic, monosaccharides must be taken up into the intestinal epithelial cell by active transport. An apical symport transports one sugar into the cell, allowing sodium to flow in down its large concentration gradient. The large sodium concentration gradient is created by constant activity of sodium-potassium ATPases on the basolateral surface of the cell, which are pumping sodium out. As secondary active transport continues to pack the epithelial cell with monosaccharides, their concentration gets quite high. Now, there's a concentration gradient driving them out of the cell into nearby capillaries. This movement occurs by facilitated diffusion at the basolateral surface of the cell. In the bloodstream, sugars dissolve into the plasma as it flows into the hepatic portal vein towards the liver. The liver takes up some of the sugars and begins to store them or use their energy. Nonetheless, the blood sugar level increases rather suddenly. When the pancreas is exposed to elevated blood glucose levels, the beta cells of the islet of Langerhans secrete insulin, The insulin causes many different cells, like liver, muscle, nerve, and fat cells, to take up, utilize, and store glucose. Soon, the blood sugar level returns to normal and you're ready for dessert. Now let's talk about proteins. So you eat a big steak. What happens? Well, in your mouth, the steak is ground and mixed with saliva. No digestion of protein occurs in the mouth. A bolus is formed and you swallow. Churning of the stomach mixes the steak with acid, mucus, and enzymes. The low pH kills microorganisms and causes many peptide bonds to hydrolyze. Activated pepsin also attacks polypeptides, breaking them into fragments. Chyme is gradually released into the duodenum. Chyme in the duodenum causes duodenal epithelial cells to secrete cholecystokinin and secretin. As a result, the gallbladder releases concentrated bile, and the pancreas secretes a basic solution of bicarbonate plus other zymogens. In the gut, trypsinogen is activated to trypsin by enteropeptidase. Trypsin then activates the other zymogens like chymotrypsinogen and carboxypeptidase. The activated proteases then go to work on polypeptides from the steak until all that's left are dipeptides and tripeptides. These are hydrolyzed by brush border peptidases. Amino acid absorption is similar to monosaccharide absorption. A secondary active transporter, or a symporter, specific to each amino acid couples the uptake of amino acid to the entrance of sodium into the cell. And when their concentration is high inside the cell, they passively diffuse out of the cell into the capillaries nearby. Like with carbohydrates, the amino acids end up in the liver where they're catabolized for energy or used in synthesis. Now moving on to fats. So you eat a bunch of ice cream, right? The almost pure triglycerides melt in your mouth and are swallowed. The stomach's churning mixes the triglycerides with acid and mucus to some extent, but because they're extremely hydrophobic, they end up floating on top. Eventually, they are emptied into the duodenum, where they stimulate cholecystokinin release into the bloodstream. Then the pancreas sends enzymes into the gut via the sphincter of Oddi. But there is a problem. Since they are organized into huge hydrophobic droplets, pancreatic lipase, which is water-soluble, cannot digest these fats. But cholecystokinin stimulates gallbladder contraction, which sends bile down. Bile comes from the bile duct into the duodenum, and this emulsifies the lipids from the ice cream, forming tiny micelles. The pancreatic lipase can now go to work. It hydrolyzes triglycerides to monoglycerides plus free fatty acids. These move into intestinal epithelial cells by simple diffusion, which they are able to do thanks to their greasy hydrophobicity and small size. Once inside, they are converted back to triglycerides, which are packaged into chylomicrons. These are large particles composed of fats and proteins which are designed to transport fats in the bloodstream. Now, as I talked about before, the chylomicrons do not enter intestinal blood capillaries. Instead, they enter tiny lymphatic capillaries known as lacteals. These merge to larger lymphatic vessels, which eventually empty into a large vein near the heart. And so a few minutes after your ice cream, huge amounts of fat are released from the thoracic duct directly into the bloodstream. Your plasma attains a milky yellow color, which is easily noticeable when a blood sample is taken. The term for milky plasma is lipemia, meaning fat in the blood. The chylomicrons circulate throughout the body and are gradually whittled away by removal of fat. In particular, adipose and liver tissues contain the enzyme lipoprotein lipase, which hydrolyzes chylomicron triglycerides into monoglycerides and free fatty acids. These diffuse into adipocytes and liver cells, where they are remade into triglycerides and then stored. That's it for today. Next time we'll go over some practice questions.